Hi, this is Sharmin here. In this video, I'm going to teach you biology, that is for Cambridge AS level, that is sodium potassium pump. Alright, so let's get started. So what is sodium potassium pump? So we know that diffusion, osmosis. If you're not watching that, you go back my another video and watch that and come back. So this is a sodium potassium pump. So the membrane, that is the cell membrane, protein, that is the membrane protein that moves sodium ions out of the cell and potassium ions into it using ATP. So I'll give you details about that. So this is a cell membrane. All right, so this is a cell membrane, and this is an extracellular space, and this is intracellular space. And here you see that is the sodium, and this is a potassium, and this is a potassium diffusion, sodium potassium pump, and this is a sodium diffusion, and this is a potassium diffusion. So that means sodium potassium pump. So how is work? Is go inside and go outside. How this work? So sodium potassium pumps are found where we found it cell surface membranes, where all animal cells. So in most cells they run all the time, and it is estimated that on average they are how much? 30% of cells energy and 70% in nerve cells. So that means it's 100% now. So this is a plasma membrane. You need to draw a diagram and label it. It can help you guys to understand. This is a sodium and this is a potassium. So extracellular, this is, I already given the information that is a phosphate here. So sodium potassium pump system moves sodium and potassium against large concentration gradient. So substance, that is the molecules, pass in and out of the cell as tiny particles, that is called molecules. So molecules in liquid and gases are not that is the never still. They keep moving and bumping into each other all the time. So and they produce energy, that is ATP, adenosine triphosphate, that is, and here is a ADP, that is adenosine diphosphate. So what's the process here? Here is a, that is the animal cells move of the molecules, so they mix in, that is called diffusion, that is the process diffusion, and molecules diffuse from where they are plentiful to where they are less plentiful. And in other words, molecule diffuse down a concentration gradient. So this is a, guys, this is, you see the concentration gradient, here is a positive and here is a negative sodium, and this is a potassium. So let's go details. So primary active transport. So active transport, here is ATP driven sodium potassium pump stores energy by creating a steep concentration gradient for sodium ion entry into the cell, and this is a cytoplasm. So sodium, that is transport leads glucose from extracellular here, and here this is releases glucose into the cytoplasm. And secondary active transport, that is the sodium diffuses back across the membrane through a membrane that is transporter protein, and it drives glucose. What's it drives? It drives glucose against its wire. You must remember that is the concentration gradient. So here you see that diagram, that is the, it moves 2 potassium ion, this is a 2 potassium ion, it moves where? It moves into cells where potassium levels are high. Here is a potassium levels are high, and pumps 3 sodium ion, this is a 3 sodium ion, it moves outside the cell into the extracellular cell, and it pumps 3 sodium ions out of the cell. So this is out of the cell, and that is the potential diffuse maintain, that is the across membrane. This is a membrane, and this is the inside. So the role of sodium potassium pump is to pump 3 sodium ion, I already given 3 sodium ions out of the cells at the same time as allowing 2 potassium ions into at the same time as allowing 2, how many potassium ion here? 2 potassium ion into the cell for each ATP molecule used. Each ATP molecules used. So ions are both positively charged. So the net result is the inside of the cell becomes more negative than the outside. That is the potential difference is created across the membrane. So what is active transport? So active transport is the movement of molecules or ions, that is through transport proteins across a cell membrane and against their what concentration gradient using energy from ATP. That is called active transport. Let's go next. Here is a one question, that is the diagram. We need to see the diagram. So here is a diagram, guys. So diagram shows part of a membrane containing a channel protein. Part of the protein molecule is shaded. So first you see the diagram. So this is a here. What's the name of this part? This one. What's the name of part B, and what's the name of part C? Pause the video, think about that and figure out. So here is identify the parts level A, B, C. So we know that A, this is a phosphate head, alright, that is phosphate head of phospholipid, and this is B, is a fatty acid tail. This is a fatty acid tail. This part is fatty acid tail of phospholipids, and number C, that is phospholipid bilayer, that is the membrane. I already given the information, that is the membrane, already you see that. So what is phospholipid? So phospholipid is a substance. This is a one kind of substance. So what kind of substance is this? So phospholipid, a substance, that is the molecules are made up of glycerol molecule, 2 fatty acids. How many fatty acids? 2 fatty acid and a phosphate group. Bilayer of phospholipids forms the basic structure of all cell membrane, that is phospholipid. So this is all about the head, that is a phospholipid head, this part, and this is a phospholipid tail here, and this is a bilayer. This is a whole thing, or membrane, phospholipid membrane, or you can say bilayer or membrane. Question number B. For each of the following state whether the component is hydrophilic or hydrophobic. Each of the following state whether the component, here is a component, is this hydrophobic or hydrophilic. So here is a this part, this is a hydrophilic, and B, this is a hydrophobic, and dark shaded area, this is dark shaded of protein, that is hydrophobic, and light shaded, that is hydrophilic. So I'm going to details about that. I will come back again this answer. So number C, explain how ions would move through the channel protein. How ions move through the channel protein. Number B, state 2 features that the channel proteins and carrier proteins of membranes have in common. channel proteins and carrier proteins of membranes have in common. Number E, state two features that the channel proteins and carrier proteins of membrane have in common and calculate the magnification of the drawing. Show your work. So you just pause the video, try to figure out. Alright, so carrier protein is a type of membrane protein which change shape to allow the passage into or out of the cell of specific ions or molecules by facilitated diffusion, or you can say active transport. So I'll already give you answer that what is the active transport, what is the facilitated diffusion. So carrier protein, what is carrier protein? That is a type of membrane protein, you must remember that, and we change shape to allow. It can change shape to allow. And how about channel protein? What do you think? This is a carrier protein, you know that, but how about channel protein? So channel protein also membrane protein of fixed shape which has a water filled pore through which selected hydrophilic ions or molecules can pass. See, facilitated diffusion, that is the channel protein. So I will give you the answer, guys. Here is a number. See, ions move by diffusion. So diffusion, that is a process, guys. So diffusion, that is the net movement of molecules, or you can say ions from a region to higher concentration. Here is maybe one ion is higher concentration to go is lower concentration. So that is called diffusion, result of the random movement of particles. So this is the process diffusion. So channel has a shape which is specific of a particular ions, and channel or hydrophilic and water filled allow movement of polar substance, and ions move down concentration gradient. That is already given, same thing here. And D, that is two features that channel protein and carrier proteins have in common. So number one, that is intrinsic proteins, yes, have a specific shape, yes. Number E, that is the state two features that the channel protein and carrier protein have in common. This is the same two features that have channel protein and carrier proteins of membranes have in the same question again. Calculate the magnification, that is, this is the channel protein have fixed shape and carrier protein have variable shape. So this is a difference. Maybe I made here membranes have is uncommon, not as a common. What is uncommon? That is their fixed shape, and carrier protein have variable shape. So that is the difference. So this is a mistakes, sorry guys. And number F, calculate the magnification of the drawing. Show your working. So here is a F number, that is C, measured in millimeter. So this is already I made one video, that is how you calculate the magnification, that is M equal to I by A. M equal to, this is the formula, you must remember that, I divided by A. So what's the I here is a C, that is the width of C divided by 7. So here is accept millimeter. If you just change it, and answer will be nanometer. So you just calculate and find it. Here is this shape is 7 nanometer. This is, we already, that is a 7 nanometer thick cell membrane. Alright, so is there any question more? So what is phospholipid? I am writing here what is phospholipid. That is the substance whose molecules made up of glycerol molecules, two fatty acids, and a phosphate group, a bilayer of phospholipid form the basic structure of cell membrane, that is phospholipid. So here is a phospholipid tail. So you see that this is a phospholipid tail here, and this is hydrophilic heads. This is a hydrophilic heads. So phospholipid tails point inwards and facing each other and forming a non-polar hydrophobic interior, and phospholipid heads, that is, you already see, this is that is phase the aqueous, that is the water containing medium that surrounds the membrane. So yeah, that's all for today. Thank you for watching, guys. Good luck your biology test. Yes, thank you so much for watching my video.